Objective: to learn the process of respiration as it occurs in plants and animals. All living organisms take in the oxygen that is necessary for the oxidation of glucose through a process known as respiration. Unlike animals, plants have no specialized organs or tissues for respiration. Instead, the surfaces of leaves and stems are filled with minute pores known as stomata. Oxygen from the atmosphere is absorbed through the stomata before diffusing into the spongy mesophyll tissue. The cells of the mesophyll contain mitochondria, where oxygen will react with glucose in a biochemical process known as Krebs cycle. During this process, there will be a release of energy, carbon dioxide, and water vapor. While the energy will be utilized by the plant, the carbon dioxide and water vapor will diffuse out through the stomata. Now let's see the difference in the respiratory system of animals using the human respiratory system as an example. The human respiratory system consists of the nose, pharynx, larynx, trachea, bronchi, and lungs. The nose is the most external organ and is the primary entry point for air moving into respiratory system. The pharynx is a common passageway for both air and food. Connecting the pharynx to the trachea is the larynx, more commonly known as the sound box. The trachea is a tubular structure that extends downward into the thoracic cavity. Splits into right and left bronchi. Each bronchi will lead to a lung where they will divide further into bronchioles and alveolar ducts, which terminate as alveoli. Alveoli are tiny grape-like sacs in which gas exchange takes place. The lungs are housed on either side of heart inside the thoracic cavity. They are divided into lobes, with the right lung having 3 lobes and the left lung having only 2. Mechanism of breathing. The diaphragm is a sheet of muscles lying across the bottom of the chest cavity. When the diaphragm contracts, the volume of the thoracic cavity will increase and air pressure inside the lungs will decrease. Oxygen-rich air will be pulled into the lungs through the nose in the process known as inspiration. When the diaphragm relaxes, the volume of the thoracic cavity will decrease and the air pressure inside the lungs will increase. Air with carbon dioxide will be pumped out of the lungs. This is what's known as expiration. Summary. All living organisms respire in order to release energy from glucose. Oxygen enters a plant through the stomata and reacts with glucose in the mitochondria of the cells. Animals have specialized organs such as lungs for respiration.
Lymphatic system. Objective: to learn the structure and functions of the human lymphatic system. Within your body, there is a system that functions largely as a drainage network, helping to keep fluids balanced, while also helping to fight against infections. This network, the lymphatic system, works with the circulatory system. Within the lymphatic system, blood enters the tissues through capillaries. At the cellular level, blood pressure causes fluid to pass through the thin capillary vessel walls to become tissue fluid. Tissue fluid differs from the blood plasma in that it contains white blood cells, also known as leukocytes. Most of this tissue fluid returns to the capillary vessels through venules. However, a small amount will not return to the blood capillary system. This part is a clear fluid containing white blood cells and is known as lymph. It will enter the lymph capillaries, which begin as blind-ended tubes that are no more than a single cell in thickness. In addition to carrying lymph, the lymphatic system has additional functions. In the small intestine, lymphatic vessels known as lacteals absorb fatty acids and transport fat into the circulatory system. It produces immune responses through the production of lymphocytes and monocytes. Lymphatic capillaries unite to form collecting vessels, which move along the arteries and veins before emptying lymph into one of the many lymph nodes found in the body. These nodes are small bean-shaped enlargements found at various points, including your neck, armpits, groin, and the walls of the intestines. There are about 600 to 700 lymph nodes in the human body. These nodes produce lymphocytes, which are similar to white blood cells. When microorganisms become trapped in the lymph nodes, the glands will swell. Summary: the lymphatic system assists in the circulating of body fluids and helps defend the body against disease-causing agents. Structure of nephron. Objective: to learn about the structure of a nephron in the kidneys. Nephrons are the functional units of the kidney. Not only they are responsible for eliminating waste products from your body through the formation of urine, a human kidney contains about 1 million nephrons. Each nephron is a long, fine tubule, about 30 to 55 millimeters long, one end of which is closed and folded with a double-walled cup-like structure known as the Bowman's capsule. This capsule is located in the renal cortex. Inside the Bowman's capsule is a cluster of microscopic capillaries known as the glomerulus. Together, the Bowman's capsule and the glomerulus are known as the renal corpuscle or sometimes a Malpighian body. Blood enters the glomerulus through renal arterioles known as afferent arterioles and, once filtered, is carried back to the bloodstream through efferent arterioles. The Bowman's capsule continues into the proximal convoluted tubule, which then continues into the renal medulla as a distinct loop known as the loop of Henle. The loop consists of two parallel limbs, one of which is descending and the other of which is ascending. The ascending limb travels into the renal cortex, at which point it dilates into a distal convoluted tubule. At the end of this tubule, a short collecting tubule is present. Several collecting tubules open into a common collecting duct, and several collecting ducts will join to form the ducts of Bellini, which then open into the renal pelvis. In each nephron, blood from the glomerulus filters water and certain dissolved substances into the space of the Bowman's capsule. The filtrate then enters the renal tubules, allowing the proximal tubule to reabsorb water, salts, glucose, and amino acids, which helps to maintain electrolyte levels in the body. The limbs of Henle's loop are permeable to ions, and the distal tubule will absorb certain ions. Urine, the resultant fluid once everything has been through the process of filtration and reabsorption, passes from the distal tubule and into the collecting ducts. It then passes from the collecting ducts through the drainage system of the kidney, through the ureters, and into the bladder for expulsion via urination. Summary: nephrons are the functional units of the kidney and filter the blood. Each nephron consists of the Bowman's capsule and glomerulus, which together are known as Malpighian body. The renal tubules include the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting tubule. The ureters carry urine from the kidneys to the urinary bladder.
Growth regulators in plants. Objective: to learn the 5 primary growth regulators in plants and their roles in the plant's growth. Plant growth regulators, sometimes referred to as phytohormones, are naturally occurring chemicals that coordinate the growth and movement of plants. Although these organic substances are produced in minute quantities, they have profound effects on plants. The 5 most common growth regulators are auxin, cytokinin, gibberellin, abscisic acid, and ethylene. Auxins are produced in the tips of the shoots and roots, and growth is dependent on the amount produced. Phototropism and geotropism in plants is due to auxins. A plant kept in partial shade will exhibit differential growth due to auxins. The shaded side of the shoot will produce more auxins, causing the cells to increase in number, which results in an elongation and bending of the shoot towards the light. In roots, the effect is the opposite. When partially shaded, that side will produce more auxins, and the roots will grow away from the light. Cytokinins are synthesized in root tips, as well as in developing fruits and seeds, and are then transported to other parts of the plant. They stimulate cell division and shoot growth. They also stimulate the growth of lateral buds, cause dormant seeds to germinate, and delay leaf senescence. In other words, the yellowing of a leaf. Gibberellins are produced by the roots and young leaves of plants. They stimulate stem elongation and delay fruit maturity and senescence in lemons and oranges. Abscisic acid, also referred to as ABA, slows down the growth of plants. In seeds, it causes dormancy, ensuring the seed will germinate only when conditions are favorable. The production of abscisic acid increases in the leaves at the onset of drought, causing the leaves to wilt as a means of conserving water. Ethylene is a gaseous hormone produced in all plants. It promotes the ripening of fruit and stimulates fruit and leaf abscission, in other words, the shedding of fruit and leaves. Synthesized plant growth regulators are widely used in modern agriculture and horticulture. Summary: plant growth regulators control and influence cell division, growth, germination, senescence, abscission, and fruit ripening. The common biological growth regulators are auxin, cytokinin, gibberellin, abscisic acid, and ethylene. Synthetic growth regulators are widely used in agriculture and horticulture.
Plant responses. Tropisms. Plants respond in various ways to changes in their surroundings. Their responses to these external stimuli are what we refer to as tropisms. Tropisms can be one of several types: phototropism, hydrotropism, gravitropism, or thigmotropism. Phototropism is a plant's response to light. This can be observed simply by placing a potted plant near a window. After a few days, the shoot will have grown towards the light source instead of growing straight up. This bending or growing of a stem towards the light is referred to as positive phototropism. The roots, however, will grow away from the light, exhibiting negative phototropism. Geotropism is the response of plants to gravity. The roots of a plant will always grow down, displaying positive geotropism. The stem of the same plant will grow upward against the gravitational pull, exhibiting negative geotropism. Hydrotropism is the response of a plant to the source of water. Plants always exhibit positive hydrotropism, growing towards a source of water. Thigmotropism is a plant's response to touch. The best example is the mimosa plant, commonly named the touch-me-not. When touched, the leaves will close, returning to their original position a few minutes later. Summary: Phototropism is the response of plants to light. Geotropism is the response of plants to gravity. Hydrotropism is the response of plants in relation to the source of water. Thigmotropism is the response of plants to touch.
Structure of neuron. Objective: to learn the structure of a neuron. We tend to think of the nervous system primarily as having to do with stimuli response, but it also works to control and coordinate the functions of the body. The nervous system is composed of billions of cells known as neurons. These tiny cells carry the messages of the body in the form of electrical signals known as impulses. Typically, there are 4 parts in neuron: the cell body, the dendrites, an axon, and the axon terminals. The enlarged part of the neuron is the cell body, sometimes referred to as the soma. This is where both the cytoplasm and the nucleus can be found, and the metabolic activity will occur here. Extending from the cell body are several short, branched extensions known as dendrites. These receive the neural impulses from receptors or from adjoining neurons, which allows them to communicate with nearby cells. It is only dendrites which carry impulses toward the cell body. Each neuron has one long, thicker extension known as the axon extending from the cell body. Axons carry the impulses away from the cell body and to the dendrites of other neurons. A fatty white coat known as a myelin sheath usually covers longer axons. The sheath serves to insulate the axon. However, it is not continuous along its length. There are many gaps known as the nodes of Ranvier. These gaps allow electrical impulses to jump from one node to the next, thus increasing the speed of its transmission. Axons end in short branches with swollen tips known as axon terminals or terminal buttons. At the tip of the axon terminals are synaptic vesicles which help to transmit impulses between axon terminals and dendrites along a tiny gap known as the synapse. Together, axons and dendrites form bundles of nerve fibers commonly referred to as nerves. Clusters of nerves are known as ganglia. Summary. A neuron is the functional unit of the nervous system. Neurons are composed of a cell body, several dendrites, and a long axon. Axons end in axon terminals, which is where the impulse is transmitted to the dendrite of another neuron.
Reflex action. Objective: to learn the basics of reflex action and the path the impulses travel as they move through the nervous system. In the nervous system, reflex actions are ones which have an automatic and quick response to a stimulus. To see a simple reflex action, when a burning candle is brought near the hand instantly moves away from the flame. Such quick responses generally do not come from the brain. Instead, they arise from the spinal cord. Using our example of the candle, let's see how reflex action actually works. The stimulus, in this case, the heat from the burning candle, will be sensed by the heat receptors in the skin of your hand. The stimulus will trigger an impulse in a sensory neuron which, in turn, transmits the message to the spinal cord. In the spinal cord, the impulse will pass through an interneuron and cross over to the motor neuron. The impulse is then carried to the effector, which, in this case, is the muscle in your arm. Instantly, the muscle will contract and withdraw the hand in response to the stimulus. This complete reflex action will happen so quickly that you'll pull your hand away without thinking about it. Because the path traveled by the reflex action from the sensory neuron to the motor neuron represents an arc, the action is often referred to as a reflex arc. In our example of the candle, the controlling center of the reflex arc is the spinal cord. Therefore, it is a spinal reflex. Reflex action canals occur in the brain, in which case it is known as a cerebral reflex. A common example of cerebral reflexes can be seen in the eyes. When a bright light is shined into your eye, particularly if the surrounding environment is dimly lit, the circular muscles of the iris will contract. Consequently, your pupil will become smaller, reducing the amount of light that is able to enter the eye. Reflex actions can be categorized as either somatic or visceral. Somatic reflexes involve the organs of the soma, referred to as the body. Such organs include skeletal muscles. Visceral reflexes involve visceral organs such as glands and smooth muscles. Summary: reflex action is an instant, automatic response from the nervous system to a stimulus. The path taken by the impulse of the reflex action is known as the reflex arc. Spinal reflexes are processed by the spinal cord. Cerebral reflexes are processed by the brain. Somatic reflexes involve skeletal muscles. Visceral reflexes involve smooth muscles and glands.
Sexual reproduction. Objective: to learn the traits and methods of sexual reproduction. Reproduction is a characteristic of all living organisms and is necessary for the continuation of life. Reproduction can be of two types, either asexual or sexual. In this session, we will focus only on the sexual reproduction. Sexual reproduction is common among higher organisms and involves male and female sex cells known as gametes. The most common example of sexual reproduction is found in flowering plants. The male reproductive part is the stamen, which produces male gametes called pollen inside the anther. The female reproductive part is the ovary, with the female gametes, which are called ovules. In flowering plants, the male pollen and female ovule will unite to produce a zygote, which will eventually form the seed that will develop into a plant. The gametes are produced through meiosis. During meiosis, chromosomes will fuse and separate, during which time genetic changes will occur. After the meiosis, the new gametes will have half the number of chromosomes and different genetic characteristics than their parents. When such gametes fuse, the zygote will have a different genetic arrangement, producing offspring that will be a little bit different from the parents. Summary: sexual reproduction is a process involving male and female organisms. Sexual reproduction includes the formation of gametes that fuse into a zygote. The offspring produced by sexual reproduction will have genetic variations. Asexual reproduction. Objective: to learn the traits and methods of asexual reproduction. Reproduction is a characteristic of all living organisms and is necessary for the continuation of life. Reproduction can be one of two types, either asexual or sexual. In this session, we will concentrate only on forms of asexual reproduction. Asexual reproduction is common among single-celled organisms such as amoeba, paramecium, and bacteria. Amoeba reproduces asexually through binary fission. With this method, the nucleus will divide by mitosis, a process that produces two daughter cells that have the same number of chromosomes as the parent cell. After the nuclear division, the cytoplasm will divide into two equal parts, which will eventually become two amoebae. Budding is a second type of asexual reproduction. In yeast, the cell will develop a small bulge. The cytoplasm and the divided nucleus will move into this bulge, known as a bud. The bud will then detach from the parent cell and will grow until it is capable of budding again. In the multicellular organism, Hydra, budding is a common mode of reproduction. A group of cells will bulge out as a bud, which will later separate from the parent. Spore formation is another form of asexual reproduction. Some bacteria, ferns, and fungi, such as mucor, commonly called a pin mold, will reproduce minute, thick-walled structures known as spores. The organism will release a huge number of spores, which will spread out and develop into adults. Summary: asexual reproduction is a process involving a single cell or organism. Forms of asexual reproduction include binary fission, budding, and spore formation. Tissue culture. Objective: to learn about the process of tissue culture. Tissue culture is a technique of producing exact copies of plants from the tissue of a donor organism. In this session, we will learn about the process of tissue culture in plants. The first step in tissue culture is to isolate the tissue from a selected plant, called an explant. The tissue can be a small piece of a root, stem, leaf, flower, or even a single cell. The tissue is placed in a petri dish containing growth medium. The medium is usually agar gel with dissolved nutrients and hormones required for the plant growth. The petri dish is left under controlled light and temperature. In a few weeks, the cells grow rapidly into small masses of tissue, called callus. Generally, green shoots develop from the callus within 3 weeks. After the shoots grow to observable length, root-growing hormone is added to induce rooting. The roots develop within 6 weeks to form complete plantlets. The small plantlets with roots are transferred to a potting medium in potting trays, where they develop into plants. Tissue culture can produce thousands of plants with exact characters in a short period. Summary: tissue culture is a form of asexual propagation of plants in a laboratory. Through tissue culture, plants with desired characters can be grown quickly and in large numbers. Types of pollination. Objective: to learn the types of pollination. Pollination is the process during which the pollen grains are transferred from the anther to the stigma of flowers. Pollination is essential in the production of fruit and seed. To understand the process, let's first look at the reproductive parts of a flower. A flower is made up of 4 concentric whorls: the calyx, the corolla, the androecium, and the gynoecium. The calyx is the outermost whorl and is made up of leaf-like structures called sepals. The next floral whorl, the corolla, is made of the familiar colorful, delicate structures called petals. It's this layer that attracts insects, such as bees, for the purpose of pollination. The plant's male reproductive organ is the androecium, which is comprised of stamens. Each stamen is a thin, long filament on which the anthers are present. These anthers contain pollen. As they mature, they will split open and release pollen grains into the air. The gynoecium, or pistil, is the flower's female reproductive organ. This organ contains the swollen ovary at the base, a long tubular structure known as the style, and a sticky tip called the stigma. Now that we know the basic parts that are involved, let's look at the types of pollination. The transfer of pollen grains from the stamen to the stigma of the same flower is known as self-pollination. The transfer of pollen from the stamen of one flower to the stigma of another plant within the same species is known as cross-pollination. In flowers such as beans and tomato, pollination usually does not require pollinators or agents of pollination. Cross-pollination, on the other hand, requires pollinators such as a honey bee. When this little pollinator lands on a sunflower to gather nectar, its legs will be dusted with pollen grains. As the bee travels to another flower, it will leave behind some of the pollen when it stops, and this will be collected by the stigma. Eventually, the male pollen will unite with the female ovule during fertilization, and the ovule will become a seed. Besides insects, wind, water, and other animals such as bats also help in cross-pollination. Summary: pollination is the process during which pollen grains are transferred from the anther to the stigma of flowers. The transfer of pollen grains from the stamen to the stigma of the same flower is known as self-pollination. The transfer of pollen from the stamen of one flower to the stigma of another flower of another plant within the same species is known as cross-pollination. Wind, water, insects, and animals are agents of pollination. Monohybrid cross. Objective: to understand the inheritance mechanism of a monohybrid cross. A monohybrid cross is a cross between two parents that breed true for different forms of a single trait. During the 1860s, Gregor Mendel, an Austrian monk, proposed the basic rules of inheritance. He began his experimentation with pea plants that bred true for a single characteristic, seed color. Because there is only one characteristic which is being bred, it is known as a monohybrid cross. Mendel bred peas with yellow seed coats for a few generations to ensure plants in subsequent generations would have yellow seeds. Similarly, he self-pollinated pea plants with green seed coloring, so all plants in subsequent generations would produce a green seed color. He then cross-pollinated the parental plants, sometimes referred to as the parental generation, of yellow seeds with the plants of green seeds. In the F1 or filial generation, each of the plants yielded seeds with yellow color. Next, Mendel crossed the seeds from the F1 generation to produce the second filial or F2 generation. In the F2 generation, the plants with yellow seeds outnumbered green seed plants 3-1. Now, let's examine the inheritance mechanism of both phenotypes, which are the visible external characteristics, and genotypes, which is the cell's genetic makeup. Each trait is controlled by a pair of hereditary units we now refer to as genes, with one unit originating from each parent. The alleles, more commonly known as forms, of the gene for seed color would be the same in true-bred plants. These will be represented by YY and YY for the parent plants. Crossbreeding YY with YY plants produces YY plants in the F1 generation. This can be illustrated through the use of the Punnett square a diagram which is used to predict all gene combinations in a cross between two parents. The F1 plants receive a Y allele from the yellow seed parent and a Y allele from the green seed parent. Although the traits for both colors are in the genes, the plants will exhibit only one characteristic, the yellow seed color. The F1 plants have yellow seeds because the Y allele is the expressed trait, meaning it will dominate over the Y allele. Thus, the Y or yellow colored seed allele is referred to as dominant, while Y or green colored seed alleles are referred to as recessive. The gametes from the F1 generation with a heterozygous genotype are matched in the Punnett square. Now let's look at the F2 generation using the Punnett square and observe the changes that occur. The square reveals that the F2 generation actually yields three kinds of plants, unlike F1 which only had the one type. In the F2 generation, 1/4 of the plants has a homozygous yellow seed color and will produce yellow seeds. 1/4 of the genotypes show a homozygous recessive nature, that is, both of the traits for green seeds. These plants will bear green-colored seeds. The remaining 1/2 of the genotype shows a heterozygous nature, as these will have both the dominant and recessive traits. The results let us see that 3/4 of the plants have a dominant yellow trait and, as such, will produce yellow seeds, and 1/4 of the plants have a recessive green trait and, as such, will produce green seeds. Thus, the Mendelian ratio for a monohybrid cross is 3:1 when looking at phenotype and 1:2:1 when looking at genotype. Summary: a monohybrid cross is based on a single trait with two alleles. The parents of a monohybrid cross are true-bred. All of the first, or F1, generation offspring will exhibit the same single trait. In the F2 generation, the progeny exhibits a 3:1 ratio of the trait. Dihybrid cross. Objective: to understand the inheritance mechanism of a dihybrid cross. A dihybrid cross is a cross between two parents that are heterozygous for two traits. Gregor Mendel, who proposed the basic rules of inheritance during the 1860s, experimented with the dihybrid cross and, based on the results, proposed the law of independent assortment. He began his experimentation using pea plants that bred true for two characteristics: seed color and seed shape. He selectively crossed pea plants until the seeds had a yellow color and a round, smooth shape for a few generations, yielding purebred plants. Similarly, he self-pollinated pea plants with a green seed color and wrinkled coat so that all plants in subsequent generations produced green and wrinkled seeds. He then cross-pollinated the parental plants, sometimes referred to as the parental generation, of yellow, round seeds with the plants of green, wrinkled seeds. In the F1 or first filial generation, all the plants yielded yellow, round seeds. Then, Mendel crossed the seeds from the F1 generation to produce the second filial or F2 generation. In the F2 generation, the plants produced yellow, round seeds, green round seeds, yellow wrinkled seeds, and green wrinkled seeds in a ratio of 9:3:3:1. Now, let's examine the inheritance mechanism of phenotypes and genotypes. This can be illustrated through the use of the Punnett square, a diagram which is used to predict all gene combinations in a cross between two parents. The F1 plants receive a YR allele from the yellow, round seed parent and a YR allele from the green, wrinkled seed parent. Although the traits for 4 characteristics are in the genes, the plants will exhibit only 2 characteristics: seeds which are yellow and round. The F1 plants have yellow, round seeds because the Y and R alleles are the expressed traits, meaning they will dominate over the Y and R alleles. Thus, the YR or yellow round seed alleles are referred to as dominant, while YR or green wrinkled seed alleles are referred to as recessive. The gametes from the F1 generation with a heterozygous genotype are matched in the Punnett square. Now let's examine the F2 generation using the Punnett square and observe the changes that occur. The square reveals that the F2 generation actually yields 4 kinds of plants. In the 16 possible combinations, 9 plants will be with yellow with round seeds, 3 will be green and round seed, 3 will be yellow and wrinkled, and 1 will be green and wrinkled. Thus, the Mendelian ratio for a dihybrid cross is 9:3:3:1. From the results of the dihybrid cross experiment, Mendel theorized the law of independent assortment, which states that each pair of alleles will independently segregate during gamete formation. Summary: a dihybrid cross is based on 2 traits with 4 alleles. The parents of a dihybrid cross are true-bred. All of the first or F1 generation offspring will exhibit dominant traits. In the F2 generation, the progeny exhibits a 9:3:3:1 ratio of the traits. The law of independent assortment states that pairs of alleles segregate into gametes randomly, independent of each other.
Inheritance of human blood groups. Objective: to learn the inheritance mechanisms of human blood groups. Human blood groups, also known as blood types, are inherited through a phenomenon known as codominance. With codominance, both the dominant and recessive traits will occur simultaneously and without blending. Based on the reactions of antigens and 2 antibodies, human blood groups are most commonly divided into 4 types: A, B, AB, and O. These 4 groups can be expressed through the use of 3 different alleles: IA, IB, and I. IA and IB are the dominant allele, while I is recessive. IA will produce antigen A, IB will produce antigen B, and the recessive allele I will not produce any antigen. During inheritance, the two dominant alleles will not blend, but will instead be expressed in the phenotype as two different traits. An individual with IA, IB will produce both A and B antigens and will therefore possess AB blood group. Let's look at the inheritance blood groups from different sets of parents. If the gametes from both parents have dominant IA alleles, all the children will inherit an IA allele, meaning each child will have the blood group A. If the gametes from both parents have dominant IB alleles, all the children will inherit an IB allele, meaning each child will have blood group B. If both parents have recessive alleles I, all children will inherit the recessive non-antigen producing alleles. This means the blood groups of their offspring will be similar to that of their parents O. Let's look at a case in which one parent has a dominant IA allele and one recessive allele I, and the other parent has one dominant IB allele and one recessive allele I. In this inheritance, both dominant alleles will pass to 25% of the children through a co-dominant pattern, resulting in blood group AB. 25% of their children will have group A, 25% will have group B, and the remaining 25% will have both recessive alleles, group O. Summary. The human blood groups are divided into 4 types: A, B, AB, and O, based on the reactions of antigens and 2 antibodies. The 4 blood groups are expressed by the 3 different alleles IA, IB, and I. IA and IB are dominant alleles, and lowercase I is recessive. When 2 dominant traits are crossed and both are expressed individually, the phenomenon is known as codominance. Human blood groups are inherited through codominance.
Sex determination. Objective: to understand the process of sex determination. The sex of an organism is determined by specific chromosomes, called sex chromosomes. In humans and other mammals, an XY sex determination system is followed. Every cell in humans, either male or female, has 23 pairs of chromosomes. Of them, 22 pairs are homologous and are called autosomes. The remaining 2 chromosomes are called sex chromosomes. In females, the sex chromosomal pair will be X and X, while in males, it will be X and Y. In humans, the X chromosome is not the same size and shape as the Y chromosome. In males, sperm cells are produced through meiosis with haploid or half the number of chromosomes, that is, 22 autosomes and 1 sex chromosome. Therefore, in a sperm, the sex chromosome can be either X or Y. Similarly, in females, the eggs have 22 autosomes and 1 sex chromosome, X. When a sperm with X chromosome fertilizes an egg with X chromosome, the offspring will be a female with paired X chromosomes. If a sperm with Y chromosome fertilizes an egg with X chromosome, the offspring will be a male with X and Y sex chromosomes. Summary. Sex of the offspring is determined by chromosomes called sex chromosomes. In humans, X and Y chromosomes determine the sex of the offspring. Females have X, and males have X and Y chromosomes.
Evidences of evolution, homologous organs. Objective: to learn about the evidences of evolution as observed in homologous organs. Charles Darwin proposed that living organisms have been evolving on Earth for millions of years. He also put forward many evidences of an evolutionary process in the form of fossils, similarities between embryos of organisms, and some anatomical similarities. In this session, we'll take a closer look at the anatomical evidences that arise from the evolutionary process. The anatomical structures of different organisms may have similar functions, but their internal structures may differ. Take, for example, the wings of a dragonfly and a bird. Both sets of wings perform the same function of flying, but unlike a bird, the wings of a dragonfly have no bones in them. Organs with similar functions are known as analogous structures. Now, let's consider the front limbs of different animals. The wings of a bird, the front legs of a horse, the flippers of a whale, and the hands of a human. If we look inside each of these organs, we'll find that all of them have the same basic structure, but the arrangement of the bones, such as the humerus, radius, ulna, carpals, metacarpals, and phalanges will be different. Although these structures are similar, their functions are different. Wings help during flight. Legs help in running. Flippers help in swimming. And hands help with a wide variety of activities. Organs having similar anatomical structures of different organisms are known as homologous organs. These homologous structures are considered one form of evidence that vertebrates with 4 limbs have descended from a common ancestor, but have become modified in the process. Summary: homologous organs are similar in structure to other organisms' comparative parts. Homologous organs form an evidence of evolution because of the implication they must have descended from a common ancestor, but were modified in the process.
Natural selection. Objective: to learn the mechanism of natural selection and its function in the process of the evolution of organisms. Natural selection is a process occurring in nature by which populations of organisms adapt to their given circumstances and evolve. The idea, which was first proposed by the British naturalist Charles Darwin, is considered an important mechanism of evolution. According to this idea, the organisms best fitting their surrounding environment will survive and reproduce, producing offspring that are already well adapted to the environment. Nature will eliminate or weed out the ill-fit individuals. Let's see if we can get a better understanding of natural selection by looking at beetles. For this, we'll imagine that our beetle population inhabiting a particular environment has 2 variations in their physical traits. They are either brown or green in color. Because nature cannot support a high population growth, most of the individuals won't have the opportunity to reproduce successfully. In our scenario, the green beetle will be attractive to birds, who will find them to be easy prey for food. The result will be a significant decrease in the population of green beetles compared to that of the brown beetles. Over the next several years, the advantageous brown color will be inherited, with brown beetles giving rise to brown offspring, and it will become the more common, dominant color in the population. Gradually, the brown beetle population will survive and continue, while the green beetle population will either become a small minority or completely disappear. In this case, nature has selected the better-suited brown beetle and rejected the ill-suited green beetles. Summary. Natural selection is a process occurring in nature by which populations of organisms adapt to their given circumstances and evolve. Nature will select the best-suited individuals in an environment and will eliminate the ill-fit individuals.
Ecological pyramids. Objective: to learn the different types of ecological pyramids. Ecological pyramids are graphic depictions of the relationships between organisms within an ecosystem. They give a quantitative overview of the feeding relationships, the number of organisms, or the energy flow found at each trophic level. These pyramids may also be referred to as Eltonian pyramids, named after British zoologist and ecologist Charles Elton, who developed the concept in the 1920s. A pyramid of numbers illustrates the total number of individuals at each trophic level. The greatest number of organisms is found at the producer level, represented by the pyramid's base. An enormous number of leaves will be needed to feed grasshoppers, which in turn will feed a few frogs, which will form a small part of the diet of snakes. Snakes will serve as a food source for an even smaller number of hawks. The number of organisms will gradually decrease with corresponding increases in the trophic levels. A pyramid of energy represents the total amount of energy that is transferred in a community of organisms. The total amount of energy available for utilization will decrease when moving from lower to higher levels. At the pyramid's base, we will find the producers, which have the maximum energy. Consumers in the next level obtain energy by eating these producers. This second level with the consumers has a slightly lower energy level than that of the producers. Moving up the pyramid to the different levels of consumers, there will be a gradual decrease in the quantity of energy available. The least amount of available energy will be in the tertiary consumer level. On average, each feeding level contains only 10% of the energy as the one below it. Energy that is lost will typically be transformed to heat. Summary. The ecological pyramids are graphic, usually upright depictions of the relationships between organisms in an ecosystem. Energy flow within an ecosystem. Objective: to learn the energy flow within an ecosystem. Any ecosystem is made up of three main components: producers, consumers, and decomposers. All are concerned with the feeding process, the circulation of chemical elements, and the flow of energy. The sun is the main source of energy. About 1% of the sun's total energy enters producers such as plants, algae, and cyanobacteria. The remaining 90% of this solar energy will be used to evaporate water or will simply dissipate into outer space. As energy is transferred from one trophic level within the ecosystem to the next, from producers to primary consumers, about 90% of it will be lost as heat as a byproduct of respiration. Similarly, as the remaining energy flows into the next trophic level, which will be the secondary consumers, another 90% of the remaining energy will be lost as heat. As the energy is transferred from one trophic level to the other, it will continue to decrease. Decomposers, such as bacteria and fungi, break down dead materials into inorganic substances and produce heat as a byproduct. To better understand how this loss of energy works, let's take a brief look at a simple ecosystem. Suppose grass produces and has 1000 kilocalories of energy. At the herbivore level, the grass will be eaten by mice or rabbits. Primary consumers has an intake of approximately 100 kilocalories. At the next trophic level, mice and rabbits will be eaten by a snake. The secondary consumers have an intake of approximately 10 kilocalories. Tertiary consumers such as eagle will eat the snake. These consumers will have an intake of only 1 kilocalories. Summary: an ecosystem is a self-sustaining structure. It has producers, consumers, and decomposers. The energy from the sun enters the ecosystem by way of producers, and from there it flows through the trophic levels. As the energy is transferred from one trophic level to the next higher level, most of it is lost. Biomagnification. Objective: to understand the phenomenon of biomagnification. Biomagnification, also known as biological magnification or bioamplification, is an increase in the concentration of a certain substance in the bodies of organisms moving through the trophic levels of food webs. This can be seen in DDT, a now banned agricultural pesticide, as it is sprayed across a field of crops. As rain falls, the pesticide will be washed off and seep into the groundwater and can also be accumulated into any water bodies such as ponds. Although the DDT will only be present in small amounts in the pond water, 0.000001 parts per million, it can still be absorbed by the algae, 0.04 parts per million, that is also in the pond. Small fish, which consume large amounts of the algae, will begin accumulating large amounts of DDT, 0.5 parts per million. As larger fish eat several smaller fish, they will consequently accumulate an even greater amount of DDT in their tissues, 2 parts per million, through absorption. Birds consuming several of these large fish will accumulate still greater amounts of the pesticide, 25 parts per million. then a human eating several contaminated fish may be exposed to significantly higher doses of DDT, 50 parts per million. In this way, a small quantity of a toxic substance will increase in concentration through the food chain or food web of an ecosystem. Summary: biomagnification is the phenomenon of an increased accumulation and absorption of toxic substances corresponding with an increase in the trophic levels of a food chain or food web.
Ozone layer. Objective: to understand the significance of the ozone layer. Earth's atmosphere is divided into 4 layers: the troposphere, the stratosphere, the mesosphere, and the exosphere. The stratosphere contains the ozone layer. The ozone layer consists of relatively large amounts of ozone molecules. This layer protects the Earth from harmful ultraviolet radiation emitted by the sun. Ozone is made up of 3 oxygen atoms, O3, in contrast to the ordinary oxygen molecules, which consist of 2 oxygen atoms, O2. Ozone molecules form when energy from ultraviolet radiation separates the 2 atoms in an oxygen molecule. The separate oxygen atoms then recombine with oxygen molecules to form ozone. The formation and destruction of ozone happens continuously at the same rate. As a result of this equilibrium, the amount of ozone in the atmosphere remains essentially the same. Although ozone molecules are only a tiny fraction of the molecules in the atmosphere, ozone plays a vital role in shielding life from harmful effects of ultraviolet radiation from the sun. Humans release into the atmosphere many chemicals, such as chlorofluorocarbons, CFCs, that damage the ozone layer. As a result, more ultraviolet radiation reaches the Earth's surface. This happens when chlorofluorocarbons absorb UV radiation and decompose, releasing a reactive chlorine-free radical. The liberated chlorine-free radical reacts with an ozone molecule to form an oxygen molecule and a chlorine monoxide radical. The chlorine monoxide then reacts with an oxygen atom, producing chlorine radical and an oxygen molecule. The new chlorine-free radical is then able to consume another ozone molecule. The resulting chain reaction depletes the ozone layer, thereby exposing the planet to the UV radiation. Exposure to UV radiation causes sunburn, infection, premature aging of skin, and other harmful diseases such as skin cancer. Since the depletion of ozone layer is a serious issue, countries all over the world have agreed to a treaty called the Montreal Protocol banning use of chlorofluorocarbons. This treaty will reduce the production and use of ozone-destroying chemicals. Summary: ozone is a triatomic molecule consisting of 3 oxygen atoms. The ozone layer prevents harmful UV radiation from reaching the Earth's surface.
Water pollution. Objective: to learn causes, effects, and control measures of water pollution. A major portion of our planet is covered by water. Unfortunately, much of it is polluted and unfit for human use, as well as harmful to aquatic organisms that live in it. The contamination of water bodies such as lakes, streams, rivers, seas, and oceans by toxic and harmful substances is referred to as water pollution. Garbage, sewage, and industrial waste are the major pollutants of water. Domestic wastewater containing human excreta is called sewage. Untreated sewage is sometimes dumped into rivers and lakes, creating a very toxic environment. Harmful microorganisms such as bacteria, fungi, and protozoa from the sewage enter the bodies of water through this waste. When such polluted water comes in contact with our drinking water, it contaminates it and causes cholera, typhoid, diarrhea, jaundice, and dysentery. Oil refineries and industries that manufacture textiles, paper, leather, paint, and chemicals sometimes release their chemical wastes as effluents into nearby bodies of water. This outflow includes toxic chemicals such as mercury, lead, arsenic, nickel, and cadmium. These will not only cause harm to aquatic organisms, but they can also enter the human body through the food chain and harm our nervous systems. Irresponsible agricultural practices may also lead to water pollution. Toxic fertilizers and pesticides applied to crops in the field will wash off into bodies of water through runoff. Fertilizers such as nitrates and phosphates will rapidly increase algae growth. Excess growth of algae, called algal bloom, will deprive fish and other aquatic organisms from the necessary oxygen, resulting in their death. Similarly, chemical pesticides from agricultural fields may run off into nearby water and contaminate the aquatic organisms. The pesticides can enter the human body through the food chain and cause harm. Oil spills and radioactive material also pollute water, sometimes damaging the environment and ecosystems for several decades. Water pollution can be controlled by a variety of measures. Sewage should be chemically treated before dumping into the water bodies. Reduce the usage of chemical fertilizers and pesticides, or look for alternative versions that are more environmentally friendly. Industrial effluents should be treated and converted into non-toxic forms. Garbage should always be disposed of properly. Summary: the contamination of lakes, streams, rivers, seas, and oceans by toxic and harmful substances is referred to as water pollution. Garbage, sewage, and industrial waste, fertilizers, pesticides, oil spills, and radioactive material are major pollutants of water. Rainwater harvesting. Objective: to learn methods of rainwater harvesting. The collection of rainwater for future use is called rainwater harvesting. Normally, rainwater that falls to earth will seep into the ground, where it becomes groundwater. Increases in construction and road work have decreased the available ground that would allow rainwater to soak in and recharge the groundwater. In addition, the excess drawing of groundwater for domestic purposes will dilute it to dangerously low levels. Rainwater harvesting is one of the methods used to conserve water. In some areas of India, check dams or earthen embankments around the fields are built to collect rainwater and allow it to percolate into the ground as a way of recharging groundwater. However, the most widely used modern method is the rooftop or terrace rainwater harvesting.
With this method, rainwater that collects on the rooftop is directed through pipes into large underground storage tanks. Once filtered, this stored water can be used. A similar method is through the use of percolation pits. The rooftop water is directed into percolation pits dug into the ground. The pits are filled with a bottom layer of brick pieces, a middle layer of gravel, and a top layer of coarse sand. The layers of sand, gravel, and brick filter the rainwater before it moves through outlet pipes into the ground. Rainwater harvesting through percolation pits also recharges the groundwater. Summary: the collection of rainwater for future use is called rainwater harvesting. Rooftop or terrace rainwater harvesting is used to store water for domestic use or to recharge groundwater.